this module, we'll be looking at 4 topics. They are, firstly, the genetic code, the structure of DNA and how it replicates, the structure of RNA, and the role of both these nucleic acids in protein synthesis. Secondly, the cell cycle, mitosis and cloning of plants by vegetative propagation, and embryo cloning of animals. Thirdly, sexual reproduction and meiosis, and finally, applications of gene technology, genetic engineering, and gene therapy. What is a gene? Well, a gene is a length of DNA that codes for a specific polypeptide. We now know from the results of work on the Human Genome Project that a gene may code for more than one polypeptide. In fact, there is one gene that is known to code for 9 polypeptides. There are many genes on a chromosome. A chromosome is one molecule of DNA, and the place on the chromosome where the gene is positioned is called a locus. Information from the HGP indicates that there are about 25,000 genes in the human genome, and we are finding that some genes code for more than one protein. Genes interact together, and genes and proteins interact together to produce a functioning human organism. In each of our cells, except for red blood cells which do not have a nucleus, we have 23 pairs of chromosomes. In females, all 23 pairs are homologous, which means they carry the same genes at the same loci. In males, 22 pairs are homologous, but the sex chromosomes X and Y are not homologous. Of every pair of chromosomes, one has come from your mother, the maternal chromosome, and one has come from your father, the paternal chromosome. On each pair of chromosomes at the same loci are the same genes, but these genes can exist in different forms called alleles. For example, there is a gene for antigens on the surface of red blood cells, but there are 3 forms of alleles: IA, IB, and IO. Any one individual will only have 2 of these alleles, and that will determine their ABO blood group. In 1953, James Watson, Francis Crick, and Maurice Wilkins worked out the structure of DNA. DNA is a large molecule. It is a polymer made of many nucleotides. So we can say that DNA is a polynucleotide. It is a double helix. The backbones of the molecule are sugar phosphate ribbon strands, and slung across them like rungs of a ladder are the base pairs. Adenine pairs with thymine and cytosine with guanine. The sugar phosphate chains are antiparallel, which means they run in opposite directions. The backbones protect the pairs of bases, which carry the genetic code. A nucleotide consists of nitrogen-containing base, a pentose sugar, and a phosphate group. The pentose sugar is deoxyribose in DNA and ribose in RNA. There are 5 different nitrogen-containing bases, and so 5 different nucleotides. In DNA are the nitrogenous bases adenine, guanine, thymine, and cytosine, and in RNA there are bases adenine, guanine, uracil, and cytosine. Note that uracil replaces thymine. Adenine and guanine are larger and are purines. Thymine, uracil, and cytosine are smaller molecules and are pyrimidines. There are base pairing rules. A purine always pairs with a pyrimidine so that the rungs of the ladder are the same width. Hydrogen bonds hold the bases together. There are 2 hydrogen bonds between adenine and thymine and 3 between guanine and cytosine. This base pairing is known as complementary base pairing. Each chromosome consists of a molecule of DNA. When the cell replicates, the DNA copies itself and it then coils and wraps around the histone proteins. It condenses and becomes organized into visible chromosomes, each consisting of 2 chromatids. Watson and Crick could see how the DNA molecule could copy itself. They suggested that the 2 strands of the molecule could split apart, exposing unpaired nucleotide bases. New nucleotide bases could then line up opposite their complementary base and form new hydrogen bonds. The 2 new molecules would be identical to both each other and to the parent molecule. Each new molecule would contain one old strand of DNA and one new strand. This idea was later shown to be correct, and the type of replication is called semi-conservative replication, as in each new molecule, half of it is from the original molecule. DNA replicates during interphase before the cell divides. The DNA molecule unwinds and hydrogen bonds between the nitrogenous bases break so that it unzips. Helicase enzymes catalyze this. This leaves unpaired and exposed nucleotide bases. In the nucleoplasm are activated free nucleotides, nucleotides with two extra phosphate groups, for example, ATP, TTP, GTP, and CTP. The nucleotides have been obtained from food. Animal and plant food will contain cells and so will contain nucleic acids or genes. We have nuclease enzymes and nucleotidase enzymes to digest this DNA. These activated nucleotides line up along the single strands of DNA, according to complementary base pairing rules, A opposite T, T opposite A, C opposite G, and G opposite C. Sections of the ribbon chain backbone are joined using ligase enzymes. The enzyme DNA polymerase plays a major role in this replication. It joins the innermost phosphate of one nucleotide to the sugar molecule of the adjacent nucleotide. The extra phosphate group fall away and energy released is used to make bonds within the new DNA molecule. DNA polymerase also ensures that only complementary nucleotide bases pair up. Each new molecule is an exact copy of the original molecule. Thus, the genetic information can be copied and passed into new cells. In 1958, two scientists in America, Messelson and Stahl, carried out an experiment that provided evidence to support Watson and Crick's theory of semi-conservative replication. They grew bacteria, Escherichia coli, or E. coli, for many generations in a medium containing the heavy isotope of nitrogen, nitrogen-15. The nitrogen atoms have an extra neutron. They then transferred some of the bacteria to a medium containing nitrogen-14 and allowed some of them enough time, about 40 minutes, to complete one division, which means one cycle of DNA replication. Others were left for long enough to complete 2, 3, or 4 divisions. DNA was extracted from each group and the sample spun in a density-graded centrifuge containing cesium chloride. The heavier DNA containing all the nitrogen-15 would settle out near the bottom of the centrifuge tube, and the lighter DNA would settle higher up. Hybrid DNA containing one strand with nitrogen-15 and one strand with nitrogen-14 would be halfway between the other two. DNA from the bacteria that had undergone one division was hybrid DNA, as shown by the position of the DNA in the centrifuge tube. This supports the semi-conservative hypothesis, but also supports the idea of dispersive replication, that each new molecule could be made of old bits and new bits scattered randomly through the molecules. DNA from bacteria that had divided twice consisted of 50% hybrid and 50% normal. This eliminates the dispersive model, as if replication was by the dispersive method, then all the DNA would have been hybrid, but slightly lighter than the hybrid DNA of the first generation. DNA from the bacteria that had replicated 3 times consisted of 75% normal and 25% hybrid, further supporting the semi-conservative method of DNA replication. Remember, DNA contains 4 different nucleotide bases. If these bases are read in groups of 3, or triplets of bases, then there are 4 to the power of 3, or 64 different combinations. There are 20 amino acids involved in protein synthesis, so this gives more than enough triplets to code for the 20 amino acids, plus a start triplet that codes for the amino acid methionine and 3 stop triplets. This also allows there to be more than one triplet of bases for each amino acid. Methionine is the only amino acid to have only one triplet of bases coding for it, and some amino acids have 6 different triplets of bases coding for them. This means that the genetic code is degenerate. It may seem odd for there to be 64 DNA-based triplets, But consider if the base triplets were read in groups of 2. This would only produce 4 to the power of 2, or 16 different codes, and this would not be enough. The first step in the production of proteins occurs in the nucleus and is called transcription. This involves making an RNA copy of one side of a length of DNA, a gene, that can pass out of the pores in the nuclear envelope and into the cytoplasm of the cell. The gene unwinds and unzips. This involves breaking of hydrogen bonds between the nitrogenous bases and leaves unpaired nucleotide bases. The enzyme RNA polymerase binds to a region at the beginning of one DNA strand, the template strand of the gene, and moves along, allowing free activated RNA nucleotides to line up alongside their complementary DNA bases. Adenine aligns opposite thymine, guanine opposite cytosine, cytosine opposite guanine, and uracil opposite adenine. A single strand of messenger RNA, mRNA for short, is produced. It is complementary to the DNA template strand and is a copy of the DNA coding strand. The triplets of bases on mRNA are called codons. This mRNA passes out of the nucleus and goes to the ribosome. Ribosomes, if you remember, are made of ribosomal RNA and protein and consist of 2 subunits. These 2 subunits come together for the second stage of protein synthesis, translation. On the small subunit, there is enough space for 2 mRNA codons to bind and be exposed to the large subunit. The first exposed codon is always AUG. Transfer RNA or tRNA molecules have also been made in the nucleus. Each one has a binding site for a specific amino acid and an anticodon, complementary to an mRNA codon. The tRNA with the anticodon UAC carries the amino acid methionine. It will align opposite the AUG mRNA codon, and hydrogen bonds will form between the complementary bases. Another tRNA with a complementary anticodon to the second mRNA codon will align and hydrogen bonds will form between the bases. Now 2 amino acids are side by side and a peptide bond can form between them. The formation of this peptide bond is catalyzed by the enzyme peptidyl transferase in the small subunit of the ribosome. The first tRNA is released. It is now free to collect another methionine molecule, and the ribosome moves along so that codons 2 and 3 are exposed to its large subunit. Another tRNA aligns and another peptide bond is formed. The ribosome moves along again. This process is repeated until a stop codon, UAA, UAC, or UGA is reached, and the whole polypeptide chain is now assembled. Stop codons do not have corresponding tRNA molecules. The sequence of amino acids, or the primary structure, is determined by the sequence of mRNA codons, which, in turn, was determined by the sequence of DNA-based triplets. Remember from the first module, if the primary sequence of the protein is not correct, it will not fold into the correct 3D shape and will not be able to function. The whole process of protein synthesis is quick. A polypeptide is assembled in about 10 seconds and many copies of the molecule will be made. The protein in question may be a membrane channel protein, a muscle protein, an antibody, hemoglobin, collagen, or an enzyme. If it is an enzyme, it may regulate a part of another metabolic pathway and may be involved in making non-protein molecules such as lipids. You can see how important the genetic code is in controlling every reaction that goes on in cells, and therefore all our metabolic reactions. Despite the activities of DNA polymerase and DNA proofreading endonuclease enzymes, mutations do sometimes occur. Some agents, such as UV light, short wavelength ionizing radiation, such as X-rays, and certain chemicals, such as benzpyrene in cigarette tar, are mutagenic. They cause mutations. A change in the sequence of bases in DNA is known as gene mutation. One of the bases in a triplet may be substituted for another. This may not alter the protein, as due to the degenerate nature of the genetic code, the base triplet may still code for the same amino acid. However, the substitution may lead to a different amino acid being inserted at that point in the polypeptide chain, and this may change the 3D shape of the protein. Sickle cell anemia is caused by such a mutation. The 6th amino acid on the 2 polypeptide chains of the hemoglobin should be glutamic acid, which is polar. Due to a change in the DNA-based triplet, the amino acid valine replaces glutamic acid on the polypeptide chains. Valine is non-polar, and having this on the outside of the molecule makes this hemoglobin less soluble. In areas of low oxygen tension, this hemoglobin forms insoluble threads and distorts the shape of the red blood cells, making them sickled instead of biconcave discs. After many cycles of oxygenation and deoxygenation, the red blood cells become permanently sickled. They can no longer squeeze through capillaries and may block the capillaries, leading to less oxygen being delivered to the tissues. This leads to painful crises and can cause death. The sufferer has an increased risk of strokes. With sickle cell anemia, both the protein and the way that it functions have been altered. If a substitution occurs in the start triplet, then the protein will not be made. If the substitution changes a triplet into a stop triplet, then the resulting protein is truncated and will not function. Another type of mutation, deletion, involves the loss of a base pair in the DNA. As the code is still read in triplets, this leads to a frame shift and all the triplets after the mutation will be altered. A frame shift also occurs with a type of mutation called addition, when a base triplet has been added. Substitution, deletion, and addition are all examples of point mutations, changes to a single base pair.